Okay, so you are already aware of this CRISPR technology, which is clustered, regularly interspaced uh, uh, palindrome sequences, which will combine with a protein and then a guide RNA sequence, which will identify specific target sites on the basis of the homology between the target RNA and the genome that is scanning, which it is scanning, and then eventually once it finds the target sequence complementary in terms of Watson-Crick base pairing, then the protein or the enzyme, that is CAS9 enzyme, what it will do is it will do induce a double stranded break, and eventually it will initiate two kinds of what we call DNA repair mechanism. So there is a possibility that we can edit the genes. Now what exactly happens is this is the sequence of the protein of this as you can see, type 2 CRISPR RNA guided endonuclease Cas9. Now this is the amino acid sequence of the whole protein. So now what has happened is one more enzyme which is very helpful in gene editing, which has been found out, that is a cytidine deaminase. Now this is the structure of the protein. Now cytidine deaminase, what it does is it binds with a particular kind of nucleotide substrate and then can able to modify it by deamination. Okay, now you can see this, this is the inhibitor which is bound with the active site, and the active site electrical surface, if we see that the active site is completely negatively in charge. Why? Because the entity that is it is binding with, it is highly positively charged because it is a nitrogenous base. Similarly, this is one protein bound with the inhibitor. Now in the other protein, what exactly happens is this is also the active site where it is accommodating isoguanine. Now isoguanine is a oxidized product of adenine, and it binds with the active site and eventually it converts into xanthine. So here also the active site is completely red, and you can see, which is highly negatively charged. So what exactly happens is is there an ability that we can change the structure of the enzyme itself, this structure of the enzyme itself, so that we can add some more beneficial aspect in this particular endonuclease. So in order to do so, there is a toolbox which will help you to understand, which works on the principles of a probabilistic classifier method, to find out what exactly happens when we undergo some kind of modification in the amino acid sequence. So we will give a quick this is the toolbox, and we are putting it pasting the sequence. Now this is the N-terminal end of the Cas9 endonuclease, and the objective is to do some kind of editing in the enzyme itself. So before doing the real-time experiment, let us do some simulations to find out whether the protein can withstand the missense mutation, isn't it? So what we have done is we have pasted the sequence N-terminal region, and on the, let us see, on the like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The 10th amino acid is an aspartic acid. Okay, so what we will do is we will edit the 10th amino acid, okay, which is already aspartic acid, with some different kind of amino acid, that is arginine. Okay, so let us see what exactly will happen if we do this kind of protein engineering in this enzyme. Okay, which enzyme? CRISPR Cas9 endonuclease. So if we change some amino acids in the endonuclease, let us see what will happen with this particular protein. So when we submit query, so what will happen? Let us see what exactly happens. So we'll refresh it. Okay, so and we'll see the results. Okay, so let us see what exactly is the result is. So what we did, this is the result, which says that if we remove the 10th amino acid from aspartic acid to arginine, it will be probably damaging, and the score is also very high, that is 1. So what does it mean? It means that we cannot do this kind of editing. So if some scientists try to see some genetic engineering approach in order to edit the amino acid sequence of the protein to incorporate some extra activity. For example, this activity, if we want to incorporate, so we can, how we can incorporate, we can edit this native enzyme into some other functions. So how we will do this? Before doing it, we have to see whether it can withstand this particular kind of editing. So here he is saying that this particular substitution is not possible because it will be probably damaging, isn't it? So this is how we would like to conclude it.